our annual T32 Progress Seminar today, and we've got some very distinguished presenters. We'll start with our very first distinguished presenter, Costa Plakis, and he will be talking about his work that he's been doing in Dr. Bob Mack's lab, and his title is on the list.
Thank you for the introduction. Bear with me 1 second. Folks on Zoom, are you able to hear us?
Yes, we are.
Nod would be perhaps.
Yeah, are they like?
Can we hear them?
Can we? That's also a great question.
You can touch the chat real quick. We can hear you.
Oh, Paul can hear us. Great. We can kind of hear you. That's good.
Okay, good enough.
Yeah, which means we could also mute it if there's any unruly.
So tell me if this helps.
Paul, could you volunteer to talk again just so we could?
Sure. How's
Oh,
it sound?
Much better.
Perfect.
Awesome.
I'm at the optometrist. What's better, 1 or 2? Let's see. We are gonna share screen. Thank you, Greg. Share. You are screen sharing.
Next to the slider for size right there.
Here?
Over, 1 more, 2 more, there.
This one.
Yeah.
Cool. All right, let's minimize this. Christopher Petro is the host.
You go over to the.
Let's just do that. Is this okay?
Perfect, yeah.
Awesome. Folks on the Zoom, can you see my slides and can you
Yes,
still hear me?
Yes, we can see your slides.
Thank you. How do I get the laser pointer? Or do I not get the laser pointer? Sorry for the, oh, there we go, hold on. Aha, cool. Glad to be the guinea pig today. Okay, so I guess I'm starting us off. Thanks, Anna Rose. So I'm here today primarily to talk about what's been the biggest project I've contributed to thus far as part of my T32. And so thusly, my talk is named Preclinical PET Ligand Development for Imaging the Endogenous Opioid Systems. But I will touch upon a couple of other different flavors of T32 related research that I've had the privilege of being a part of since April Fool's Day of 2021 when I started. So the first third of the talk and what will take the lion's share of today's resources is the development of a mu opioid receptor non-agonist PET tracer. And that's been primarily accomplished through carbon-11 radiolabeling. I have a kind of a recent labor of love, and that's the F18 radiolabeling of what I think might be a good clinical candidate of a lead ligand that I've been exploring. And that's a direct isotopolog, so they're related. One is F19, one is F18. Towards the end of this mu opioid section, I started to do some interesting work on the development of synthetic opioid rescue agents, which I think will be interesting. Somewhere down the line, if we have time permits, I could also tell you about the K99 I'm planning to submit. And then I don't think this time will allow for any discussion about this last point, but if anyone has any questions, I'm happy to chat offline or perhaps discuss the possibility of a collaboration. And that's using PET to evaluate the changes associated with both hallucinogenic and non-hallucinogenic psychedelic treatment. But to kind of get us started, get us thinking about the problem that I'll be talking about in the next couple slides, and I'll gloss over the pharmacology since many of you are well informed in this area, but opioids are and remain the standard of care in the treatment of acute pain, where opioids, which is represented by this blue scaffold, will bind to one of the opioid receptors, and this is a class of, or these are a class of G protein coupled receptors, and they can induce either beta arrest and recruitment or G protein mediated signaling. And research primarily today is engaged with designing biased agonists, or more relevant to, I think, NIDA's mission is the development of novel displacers that combat the opioid epidemic that we still find ourselves in. So historically, you might think of these morphinan-like scaffolds. So this is morphine, this is heroin, morphine being the more commonly prescribed of the two, as being a historical driver of opioid morbidity and mortality. That's sort of changed since about 2013, where the lion's share of morbidity and mortality are arising from these synthetic opioids. So since about 2013 onward, we find ourselves in this synthetic opioid era, synthetic opioid crisis, where the potency and relative ease with which these are synthesized contributes to their pervasiveness outside of a clinical environment. So it's somewhat easy to get your hands on illicit starting materials to carry out some clandestine chemistry. And again, that's contributing towards a tremendous loss of life and also financial burdens. I think one way to kind of get at the crux of this issue is to give researchers more tools or better tools to study opioid neurobiology. And historically or classically, when I think about studying opioid neurobiology, I think about using either a PET radioligand or a tritiated ligand that you might do some in vitro studies with. So here are just a handful of well-characterized, well-described ligands. These are positron emission tomography ligands that you'd find in the literature. And generally, they fall into one of two categories. Either they are, generally speaking, non-selective, such as in the case of F18 cyclophoxy, which I could also tell you is very tricky to synthesize the relevant precursor. Carbon-11 diprenorphine is also non-selective, and AH7921 suffers from a pretty long radiosynthesis time that kind of precludes its use in studying clinical populations or being used to study fundamental opioid neurobiology. We do have something that is both potent and selective, and that comes by the name of carbon-11 carfentanil, which is so potent and so selective, you actually need a Schedule II DEA license to use it and deploy it clinically. And it follows, therefore, that carfentanil production requires stringent quality control. And I would argue that this limits the ability of researchers to study opioid neurobiology, particularly in a high throughput manner that's sort of needed to address the issues that the field is facing. To solve this, I've been inspired by a molecule eventually, or rather historically developed by GlaxoSmithKline. So here is a GSK 152-1498. If you've tuned into my previous talks, you've noticed my infatuation with this molecule, and I'm here to convince you that it's a molecule worth studying. So it's progressed through 2 phases of clinical trials. It's relatively non-toxic and safe, and the key selling point of this as a potential radioligand is that it is a non-agonist. And so it carries with it a much safer pharmacokinetic and pharmacodynamic profile, which should obviate the need for a DEA license. And I would argue will allow researchers a new tool to study opioid neurobiology in a bit more of a high throughput manner.
How does it allow the study of the active state?
Right. So carfentanil being an agonist might have or might bind to a different pool of receptors than a non-agonist. So it's thought that a non-agonist might bind the inactive and active state, whereas carfentanil kind of only binds the active state. So there are lots of really subtle and yet interesting things that I think a non-agonist PET tracer would give us. And to sort of start down this path, I did kind of what any medicinal chemist would do, and that is to make a bunch of molecules. So I designed a modular synthesis from a relatively modest number of precursors. And in 3 to 7 steps, you're able to synthesize a relatively large library of analogs, which I've turned just GSK 152 analogs or congeners, that one, allow us to tease out the structure-activity relationships, and 2, give us a handle or something that we can viably radiolabel. And so you can see from going from the parent molecule, if we introduce this methyl group, which is a viable handle for radiolabeling, you decrease the affinity of the molecule, and I'll touch upon how we measure affinity on the subsequent slide. But what you can then appreciate is we can use some of these medicinal chemistry-like tricks in switching this triazole to a benzamide, where you can sort of regain some of the affinity associated with the parent or the lead compound, as well as maintain a general improvement in the selectivity of the binding to the mu opioid receptor versus the delta and kappa opioid receptor. We use the binding affinities in conjunction with, this is a tool we just use to predict the blood-brain barrier permeability of our nascent cold molecules. So this is appropriately named the boiled egg plot. And so molecules possessing the right combination of the calculated log P, which is a measure of lipophilicity on the y-axis, as well as the correct measure of topological polar surface area, help relatively polar or non-polar a molecule is, these molecules may find themselves in the yellow of this boiled egg. And that is an area which we predict would have a high likelihood of crossing the blood-brain barrier. So again, you can kind of correlate these structural changes you make in introducing this methyl group and or switching the triazole to a benzimide to relative changes in the lipophilicity. This I often use in conjunction with CNS PET or PET MPO. PET multi-parameter optimization is just a variety of different tools that sort of allow you to triage your workflow. Because the worst thing to happen is to pick a molecule that you think will get into brain without any substantive modeling data, radiolabel it only to have no brain uptake. That's what we're trying to prevent by using this and related modeling technologies. The binding affinity we measured in collaboration with the PDSP. This is the Psychoactive Drug Screening Program at the University of North Carolina, Chapel Hill. That's under the direction of Dr. Brian Roth, who should be no stranger to the P30. He's sort of a tour de force in not just opioid pharmacology, but broadly speaking, all GPCR pharmacology. And we made use of his radioligand competition binding assays to query the relative binding affinity of these ligands, not just for the mu opioid receptor, but also selectivity over the delta and the kappa opioid receptor. And so again, what this assay measures is the relative ability of your ligand or a standard to compete off a tritiated, known and well-characterized radioligand. And that gives you a Ki value, or relative binding affinity of your ligand to the relative or to the receptor of interest. And so I've done this for the mu, the delta, and kappa opioid receptor expressing cell lines. And hopefully what we can appreciate is that these ligands that I've developed, 1A and 3A, are about equipotent in their binding as compared to morphine, while offering some degree of selectivity for their binding over the delta opioid receptor and kappa opioid receptor. All relatively important things to consider when developing or hoping to develop a mu opioid receptor specific PET radioligand. We also looked at their relative off-target liabilities. And so what we can see here and hopefully appreciate is that these iterative fluorine additions or removals play a large role in dictating whether or not these ligands will bind to targets other than what we've designed them to bind to. So we can see there's a small liability with compound 3A, where it has a predicted somewhat high binding affinity to the dopamine 4 receptor, but also, and probably more problematically, the norepinephrine and muscarinic receptor, and that we can sort of remove this off-target binding just by kind of an increase in this one lipophilicity, but through the introduction of these fluorines in the central ring of this pharmacophore. So I've demonstrated predicted blood-brain barrier permeability and relative binding affinity. The next thing I'd like to talk about is what I think is the real selling point of these molecular entities as a suitable radioligand is their non-agonist behavior. So this is the Presto Tango assay, also developed by Brian Roth and is available to NIH-funded researchers through use of the PDSP. And what this demonstrates is here we have mu, delta, and kappa opioid receptor expressing cell lines, and agonist ligands bind to and induce G protein and beta arrestin signaling. And the eventual beta arrestin 2 signaling in this assay eventually leads to the increase of relative luminescence intensity through the transcription of luciferase. So for lack of a better term, agonists turn the lights on. So DAMGO, we see a relative increase in luminescence intensity, where we don't see that for any of the ligands, or particularly this 1A ligand in the mu, delta, or kappa opioid receptor cell line. So we can confidently say that our 1A lead ligand here behaves as a non-agonist. And when you look at this in antagonist mode, and so this requires the pre-incubation here with a known agonist. So you have some basal activity, but that's not a constitutively active receptor. It's actually induced by a pre-incubated agonist ligand. And so what we measure here is the ability of 1A, or in this case, naltrexone, to more or less compete off that pre-incubated agonist ligand and decrease or shut down the relative luminescence intensity. And so what we think is happening here, or at least what one possible explanation is, that 1A can kind of compete at the binding site with the pre-incubated ligand and exhibit similar pharmacology as naltrexone, which is depending on the model, known as a mu-opioid receptor, either antagonist or in some cases inverse agonist. And this behavior is conserved in both the mu and the kappa cell lines. The delta cell lines are a little bit unique. There is no reference ligand to compare this to just yet, although I'm in conversation with the PDSP to see if that's something they're developing. But at the minimum, we can say there is no increase in relative luminescence intensity, meaning there's probably no signaling going on, and that this behavior, this non-agonist behavior, is conserved for both 1A and 3A. So again, all demonstrating non-agonist behavior, offering what we think is a safer PK or PD profile compared to known radioligands. So I went ahead and radiolabeled both 3A and 1A to make carbon-11 3A and carbon-11 1A. I'll spare you the details of the precursor synthesis, but the radiosynthesis showcases some really neat radiochemistry that was developed in part here at Penn through a collaboration with Merck and Princeton. So this is it's a mouthful, but it's a metallo-organo-photoredox reaction where you couple cyclotron-produced carbon-11 methyl iodide with more or less the aryl or alkyl bromide of your choosing. And this witch's brew of reaction contains various catalysts, co-catalysts, supporting ligands, and most interestingly, light. So the light excites your iridium catalyst, which is then enables this relatively complicated radiochemistry to be carried out. And last I checked, there aren't many cyclotrons that can do this, but it allows you to access otherwise inaccessible radiopharmacophores in kind of a neat and pretty sophisticated way. The radiochemistry or the radiochemical purity, of both of these nascent radioligands is suitable for injection. The radiosynthetic time is accomplished in under an hour for each of these, which is generally what's required when working with a short half-life isotope such as carbon-11. And the specific activity, which is the amount of radioactivity added sort of per mass unit, and so we measured that in millicuries per micromole, is about 800 for carbon-11 1A, or sorry, carbon-11 3A, and 1400 for carbon-11 1A, and that's normalized to the end of synthesis. So we were able to generate some striking images from the PET tracers that I've radiolabeled. And as a disclaimer and/or as a comparator, on the top, we have C11 carfentanil demonstrating rapid uptake in mu-opioid receptor expressing areas of the brain. The sensitivity is not quite as good for 3A or 1A, and I primarily think that's driven by the lower binding affinity. So carbon-11 carfentanil has picomolar binding affinity for the mu-opioid receptor, which I think gives it a much better binding potential, whereas 3A and 1A having a slightly lower Ki measured gives us not quite as pretty images, but definitely a more safe pharmacodynamic profile. And to date, we've done several baseline scans for carbon-11 3A, as well as some dynamic PET scans. And so these displacement studies utilize first the administration of radiotracer, followed by administration of some sort of displacement agent sometime after radiotracer administration. And I'm happy to chat more about these offline, but in the interest of time, I thought I'd kind of just keep progressing here. But all of these, it's my goal to have sort of all these wrapped up in a JMedChem manuscript and submitted ideally before I submit a K99, which we'll get there. We'll get there. So at the end of this portion of the talk, I hope I've been able to kind of convince you that this is this GSK and related congeners represent a pretty interesting and unique scaffold where we have non-agonist behavior that are amenable for radioligand development. As I alluded to, the affinity of some of the PET ligands that I've developed are a concern. And I think working forward, one way we might address that is through the direct radiolabeling of this GSK molecule directly. And so that would be what's known as an isotopolog, where you take this available. This would be an F19 isotope that's available in cold chemistry, and then hopefully replace it with an F18 radiolabel. And so the reason you might want to do that again is because this GSK lead molecule possesses higher or relatively more potent binding affinity. And I've made some headway in this. I've worked backwards through a retrosynthesis, and I've deployed some, I'll call it old school chemistry to kind of start developing this. And so this makes use of this electron withdrawing group, ortho to an aldehyde to stabilize what's known as the intermediate Meisenheimer complex. Happy to draw structures if anyone's interested in the organic chemistry later, but you can get the incorporation of this F18 radioisotope in pretty high radiochemical yield. And then the subsequent steps are just to construct this carbon-nitrogen bond through either a reductive amination or to use, there's some existing group literature that suggests this could also be accomplished through an N-alkylation. But again, happy to chat offline more if people are genuinely interested in that. But I thought it was a little dry for this group. So I'd like to now, if time permits, and I didn't set a timer, so Anna Rose, am I good? Am I bad?
Start about 4 to 5 minutes late, so
Okay, so I have like 5-ish. Yeah, okay, cool. So I'll switch gears now to talk about the use of some of these ligands as not diagnostic imaging agents, but therapeutic agents to help address the issue of a prevalent issue in the literature that's becoming more and more emergent, and this is known as the issue of re-narcotization. So this is a not so cheerful report from Morbidity and Mortality Weekly Reports, which details several patients' journeys through the emergency room. And the take-home message, or what bands all these patients together, is that they were found suffering from opioid use overdose. And traditional naloxone treatment did not work on all of them, which is quite frightening. So several patients who were alert after receiving naloxone or Narcan subsequently developed respiratory failure, which is frightening, right? So you've encountered somebody suffering from overdose, perhaps they're unconscious, suffering from respiratory distress. You do everything you're supposed to do, administer Narcan intranasally. They appear alert, but later they slip back into being unconscious, which is quite frightening. And I think a failure of the current pharmacological design. So now this is a modeling paper out of Nature Communications, which suggests that this is fentanyl, a synthetic opioid analog. It engages in a slightly different binding mechanism than canonical opioid agonists do. So many canonical opioid agonists bearing resemblance to or having that morphin scaffold can be displaced by naloxone, which again resembles that morphin scaffold, but these slightly longer and elongated pharmacophores, such as fentanyl and related synthetic opioids, can move a little bit deeper into the binding pocket where it's thought they could engage with this histidine residue, perhaps contributing to a slower off rate or certainly more potent binding affinity. So what I argue for is the use of this chemical technology we've sort of recently come into exploring and its ability to displace potent opioids. So this is a paper that CJ has really been the workhorse in getting this submitted to the European Journal of Nuclear Medicine and Molecular Imaging. But what we're demonstrating here is these are time activity curves that follow the administration of C11 carfentanil. And in figure A, this is a dynamic PET scan where you can see the uptake in the thalamus. And so this is a mu opioid receptor rich area of the brain. You can see sort of this prolonged uptake. And what we do is administer some sort of displacer, either naloxone or this GSK lead compound, at about 40 minutes, and we see kind of what happens. Does it displace it? Does it increase binding or something else? And what we see is that it looks like naloxone and GSK kind of displace C11 carfentanil, which is an excellent scaffold for a synthetic opioid at a somewhat similar rate. And that rate is somewhat enhanced compared to baseline studies. And so where I think this will eventually lead to is the ability or the perhaps some sort of grant submission where we evaluate the ability of this molecule to compete off newer or next generation synthetic opioids. So there's already kind of been some literature developed by or data published by the GlaxoSmithKline team where they show this ligand is very efficient at shunting or decreasing, in this case, metenkephalin induced signaling. And also, if you look at this, this is a GTP gamma S assay. This seems to be the only molecule, GSK, that shuts off or diminishes constitutive activity in a mu opioid receptor expressing cell line, suggesting it might not just have antagonist or non-agonist activity, but perhaps inverse agonist activity. And certainly in the literature, particularly work done by Wolfgang Sadi from Ohio State University, he's advocating for the use or discovery of molecules that are able to perhaps bind to this active state without necessarily blocking signaling. So you could still get analgesia, but also accelerating this constitutively active mu opioid receptor back down to the ground state, hopefully shutting off things such as dependence, hyperalgesia, or respiratory depression, things with very poor clinical outcomes. So that kind of wraps up the mu opioid receptor related area of my talk. And I think I'm just about at time.
So take a couple minutes to summarize it and also say how wonderful it is that you're now finishing your T32.
Yes. So it is, Anna Rose didn't pay me to say that, but she's encouraged me for the folks on Zoom to just summarize, I guess, what I'm doing next and how grateful and wonderful it has been to be part of this T32, which really has been. Yeah, it's been certainly a life change. Best thing I've done for my career is come to Penn. I think I told Anna Rose when I met her, when I was just a little baby freshman in chemistry, I met Joanna Fowler and had this pipe dream that I would, one day go and do radiochemistry. And so that's been kind of like, coming full circle, being here, doing that. And the T32 has sort of been instrumental in learning the radiochemical tools that I'll need to hopefully, great segue, complete and compete for a K99R00, which will focus on not mu opioid neurobiology, but delta opioid neurobiology. And so just as kind of a high level overview, there are delta opioid receptor specific radioligands. They suffer or have historically suffered from poor radiosynthetic reproducibility. And also they don't have great kinetics. This methylnaltrindle is developed in the 90s and doesn't reach equilibrium until greater than 90 minutes. Is not exactly compatible with a 20-minute half-life, meaning if you wanted to do a dynamic PET scan, probably isn't going to work in all honesty. And so as I've often done, I've looked in the patent literature for molecules that might serve or be able to be repurposed to find a suitable PET radioligand. And I was inspired by this. This is a somewhat local molecule developed by Trevena. They're Philly-based. And this has been through a few phases of clinicals to treat migraine, which I think is interesting and perhaps relevant to NIDA's mission in that there are circumstances where mu opioid receptor targeting therapies are prescribed off-label to treat migraine. And I think developing a delta opioid receptor radioligand would, one, enable clinicians and other scientists to more rapidly screen chemical matter that interacts selectively with the delta opioid receptor, perhaps kind of diverting the use of mu opioid selective therapies from being prescribed off-label to treat migraine. And also just the delta opioid receptor is historically very understudied. I would argue we don't have a great idea of what the neurobiology of it is just yet. And then the K99 aspect of this would be to do this via a cheminformatics method. And so as a medicinal chemist, I'm very familiar with mixing molecules at a fume hood. But what I'm less good at, and where I think I'd greatly benefit from training, is doing this using computer-assisted drug design. So I have made some ligands and then some initial structure-activity relationships. But what will enable, I think, the field to identify hits a lot quicker is through the utilization of something called an in silico similarity search. So finding relative hits that look okay. And so these have modest binding affinity and selectivity, but filtering this through either a ligand-based in silico similarity search or perhaps a structure-based search where you dock this with a G protein of interest, find select hits, cluster them, do some visual inspection. And then the nice part about both of these molecular libraries is that you could order them relatively easily, and or a lot of them are pretty easy to synthesize So easy, a humble medicinal chemist could do it. And then the idea would then be to do this via a high throughput screen, and identify some chemical matter that you can then do hit to lead optimization with, so on and so forth. So I'll stop there. Just a couple quick shout outs of people that have sort of helped me along the way. Obviously to Bob, who's been a fantastic mentor through my last 3 years here. Looking forward to kind of our continued working together, maybe more related to the U19, but hopefully not too much related to that. Tom was great when I got here. He kind of was the igniting force behind this project. CJ is great. Best I could describe her. She's a wizard that helps with a lot of our pet modeling and our screening. The cyclotron staff, Sharon Lee and Alex Schmitz, have been instrumental in my training as a radiochemist. The collaborators at the Roth Lab PDSP. Simtra, who is instrumental in getting our new instruments up and running in the cyclotron, the T32 personnel. Julie and Anna Rose, you've been great. And I've also been lucky to kind of shadow and work along the P30. So with that, hopefully I haven't taken too much time, but I'd be happy to welcome any thoughts, comments, or bitter remarks. So have at it.
First of all,
Thank you. And I will stop sharing my screen just to see if there's anything in the chat. Nope, nothing in the chat. Cool.
so, anyone here have a question, or do you want to go and look at our faces online? And see if there's people who might have a
Yeah.
question that don't have the joy of being here in the room with us? Open for questions.
We're open for questions, internet gang. I have yeah, the screen pulled up, so perhaps if you raise your hand, this might.
Process,
Go
I was
on.
really intrigued by the notion of the overdose and that naloxone doesn't really work that well, probably because of the fentanyl or the synthetic fentanyl. So did you see a difference in displacement between between naloxone and your GSK compound? I couldn't really see it.
Yeah, great question and good observation. So for folks online, the question was, is there a large or I guess significant difference between displacement in
The
these?
first graph, it's a little different.
Yeah, so here we have just dynamic PET scan showing uptake of carfentanil with administration of both naloxone and GSK. And so the take-home message is it looks like they displace carfentanil at about the same rate as evident through graph C. But what I don't know and what I think we need further investigation for is whether or not this ligand would work well in a re-narcotization model. So again, both naloxone and this GSK lead compound displace carfentanil. But what I don't know is these new ligands that have perhaps a different binding mode or look differently in the binding pocket, will those prevent rebinding that would then prevent re-narcotization. Additionally, I don't know whether or not they're truly an antagonist or inverse agonist and what role that might be in diminishing constitutive mu-opioid receptor activity back to the ground state, which might be also important for preventing respiratory depression.
And Costa, you can brag on yourself because in line with what you're talking about is the excitement of being able to actually work on better and better tracers, but that may have really interesting and unique therapeutic potential in the ways that you just described, right? It's
Yep.
not out there in the field and very, very, very greatly needed. And the need is not getting less, it's greater. The NIDA are coming down the hall.
Yes.
It's just unending. So people will be beating down your door or whoever's still there in the mock lab. I'm teasing, but basically, because these are questions that are so very important, they're not going away.
Right.
So for the imaging field, for sure, we need obviously something easier and better than carfentanil, but also for therapeutics. So
Agreed.
we're very grateful to have you and very grateful to have you as this year's first graduate of this cycle.
Thank you very much.
Good job, Ben.
So I thank you, everybody online. Hopefully that means I could exit stage left, but I'm happy to entertain discussion offline with anyone who's interested. And I'll pass the microphone to Greg. I was Greg's opening act.
Julie, did you want to?
So Greg, for those of you who may or may not know, graduated from Vanderbilt University with a PhD from Danny Windner and a lot of electrophysiology. And he joined Greg Quarter's lab 2 years ago, about
3 years ago.
3 years ago, and did some really elegant work. I don't know if that's what you're going to be talking about.
Pretty much, yeah.
With
It's
sort
the
of-
most relevant to what we're doing here on the T32.
And is currently over in CHOP, actually working on some CAR T cells and immunotherapies-ish,
Yeah, more or less.
more or less.
Okay. Thank you very much for that, Julie. Yeah, so I will get started just so we can keep this rolling along. But as Julie alluded to, this is kind of going over work that I did when I was still over at Penn. I'm at CHOP now, but this is work that I did in the lab of Greg Quarter for the majority of my postdoc over there, and kind of keeping the whole tool development theme rolling, kind of following Costa's wake here. We'll be talking about some of these new viral vectors that we designed, the suite of tools to more selectively target AAVs to mu-opioid receptor positive cell populations in the brain of research models, and hopefully something that could actually go forward to being looked into as clinical-based therapeutics for patients with opioid addiction or intractable pain disorders too. I'll preface this by saying as well that this work has already been published. It was just accepted back in September of 2023. So if anyone's interested in more, and we don't have enough time to go through everything, you can definitely look up the paper. I can give the DOI or whatever to anyone who's interested. So not needing to belabor this point, Costa already introduced it very well. Obviously, there's a big problem both within the US and then globally with these increases in pain-related and opioid addiction-based disorders. There's a large endemic, and there's a big need to find better ways to both treat these disorders, but also look for different ways to kind of do this outside of the conventional means that have been mostly in vogue for the past several decades, which usually involve using other kinds of opioid agonists or antagonists to kind of target the mu-opioid receptor selectively. But of course, it's come with a lot of drawbacks in terms of engaging opioidergic-based pharmacology, which can lead to some of the negative clinical outcomes that Costa nicely summarized at the end of his presentation there. So one of the ways that some folks have been thinking about is kind of following the wake of a lot of these new tools that have been developed over the past several years for finding ways to more directly manipulate and target the cells that kind of drive opioidergic signaling within the brain without the need to use a lot of these pharmacology-based mechanisms that, again, have several drawbacks that are associated with them. And that's principally what I was kind of involved with doing when I was working with Greg Quarter over at Penn. So again, don't really need to belabor this too much. I'm sure most people know this is kind of the main receptor that we were kind of trying to target in terms of the population of cells that express it, the mu-opioid receptor. Obviously, they're very highly expressed on cells that are involved in both pain processing, which was mostly interested to our group, but also on reward circuitry and cells that are involved in the nucleus accumbens to ventral tegmental area reward circuitry in the brain that's canonically studied in a number of different animal models of addiction as well. So this is the population that we're going after. And obviously, we also know that these are highly relevant cells for a lot of these different kinds of studies, both in the fields of addiction and pain, due to their high expression levels on a number of different regions, not only in rodent and other animal model brain structures, but also in human brain structures that have been highly associated with a number of different cognitive and reward-based tests. So here, just kind of, again, following the wake of Costa, this is a carfentanil screen showing a lot of binding affinity of a radiolabeled carfentanil molecule to a number of different very highly salient regions, such as the anterior cingulate cortex, the amygdala, brainstem structures, and other midbrain regions that are highly involved in driving a lot of critical aspects of both pain and motivated behavior. So the main question for us was, is there a better way to target and study these regions and circuits without engaging or over-engaging the activation of this reward or this opioid-based circuitry, and still find a way to kind of preserve the basal functions of the cells without really trying to drive patients or other study animals into a state where they're going into overdose or withdrawal by using too many of these more negative pharmacological compounds. So our approach is mainly, as my title kind of alluded to, was trying to find a way to leverage some of the new viral tool-based designs that have been all the rage in neuroscience for the past several years, especially at the preclinical animal model level, to more directly and selectively target a specific population of cells. In this case, the ones that express the mu-opioid receptor more selectively, and use these viruses to encode a number of different transgenes and other kinds of transgene-based cargo to allow us to more directly and selectively have control
over these mu-opioid receptor cells that should putatively be involved in driving some of the aspects of pain neurobiology and addiction neurobiology that we see both in humans and animal models that are used to kind of look at these and study these at the preclinical level. And here's just a couple of examples for anyone who's not super familiar. It might be a little esoteric, but a lot of ways that we can do this is by utilizing viruses that can selectively encode a reporter molecule, something like a green fluorescent protein, or that can make use of a number of these new tools that are these different kinds of ion channels or light activated channels or channels that are activated by selective drugs that will allow you to more directly modulate the activity of neurons, either by turning them on or off in a very reductive way of describing that with light signatures, or turning them on and off with these different kinds of agonists that have been exclusively designed to have no other kind of biological activity outside of activating these synthetic receptors. And that's principally these DREAD receptors. Again, we can thank Brian Roth, as Costa mentioned, for the development and the application of in neuroscience. So how did we go about doing this? So the main approach that we took was trying to find a genetic signature that would allow us to more exclusively drive the expression of these viruses in the select population of cells within the brain. The way that we tried to go about doing this was designing a viral construct that was exclusively targeted to the promoter regions that's known to be present in any cell that is able to produce the mu-opioid receptor on their cell membrane surface. So we designed a couple of different construct sequences with the help of our collaborators over in Carl Dijsselroth's group, principally Dr. Charu Ramakrishnan, who really spearheaded this work and started collaborating with Greg when he was still finishing up his postdoc back at Stanford. But essentially, we tried to find a couple of different genetic sequences that would more selectively recognize this entire region of the opioid promoter portion of the genome in both mice, as you could see here, and in humans as well, which are the two kind of sequences that we tried to design here. And we were able to get some nice hits. The boxed yellow area up there was the sequence that actually worked really nicely for the mouse genome. And then the first hit that Cherry tried, the first construct she designed for the human genome, actually worked really, really well. So how do you get that good? I don't know, but she definitely deserves her retirement now, which is what she just started a couple months ago. But it was really great that she could help us finish this up. And on the far right, you can kind of see this is just the basic schema of what we then decided to do. Once we developed these two constructs, it seemed to really bind very selectively, exclusively to this region of the genome that encodes the mu-opioid receptor. So we went ahead and started to design this suite of viral tools working via adeno-associated viruses to kind of drive not only this construct that recognizes this promoter region of the mu-opioid receptor in general, but also encodes a number of these transgenes. So you can kind of see a couple of them listed on the side there, but we'll talk about them in a little more detail over the next couple of slides or so. So of course, we need to test this and make sure that it worked well and was selective, obviously for the mu-opioid receptor positive cell populations. Charu did this at first in vitro by taking cultures from Wistar rats that she had in her lab. These are primary cortical cultures, and just kind of treating the plates of cells that she was growing with our virus, and then trying to see via IHC whether or not we had expression of the reporter fluorophore, in this case, enhanced yellow fluorescent protein. As you can see pretty nicely across both tests of the mouse and the human version of these viruses, we got pretty nice expression. It's not in every single cell type, so hopefully that was at least indicating that the virus worked and was able to get into neurons of interest in general. We then went and expanded this, working with mice as opposed to rats, and did more in vivo validation testing just to make sure that this virus worked selectively and was able to express in the brain. So across all these images you can see from different sections of brain from the mouse, when we injected both the human and the mouse version of this virus, we were able to see very nice restricted expression in a number of different structures that are known to more selectively express the mu-opioid receptors, or areas like the prefrontal cortex, the central nucleus of the amygdala, as well as areas like the periaqueductal gray, which is known to have a very high expression of mu-opioid receptors. And this was kind of seen across both of our viral constructs that we developed. We also wanted to make sure, again, that this was more highly selective for neurons and not other kinds of different cell types that might be in the brain. Obviously, there's a number of different glial cells that are expressed throughout the brain. While the jury is still out on whether or not they do express mu-opioid receptors, there was some kind of contention in the literature with certain groups saying that microglia, the dominant immune cell of the brain, express mu-opioid receptor on them. So this was just another round of injections and then staining that we did both for our virus and then using some antibodies that were more selective to neurons. This new N stain here and then IBA1, which is a stain for microglia. And what we were able to see is that there was really no overlap at all of our virus with the microglial cells that we were able to stain here, but pretty much the majority of the cells that were positive for our virus and the transgene there, the EYFP, pretty much always overlapped with a new N stained cell. So it seemed fairly selective to neurons, which was nice. Finally, of course, with some of this initial validation, just at the level of histology, we of course need to make sure that great that the virus expresses, but is it actually in selectively mu-opioid cells more predominantly than it would be in other cell types that don't express the mu-opioid receptor on them, or at least have the ability to express the mu-opioid receptor by having either the gene sequence in it or able to produce transcript from that. So here, this was just some basic staining using both in situ hybridization approaches as well as immunohistochemistry approaches, which you can see on the respectively in the images on the right there. So when injecting the virus, taking slices, and then staining for OPRM1 mRNA, which is the gene name for the mu-opioid receptor, we see pretty nice and selective restriction of our viral expression over cells that do have this mRNA transcript present within them, as well as when we stained for anti-Cre, in this case in a mouse line that was a transgenic animal that expressed Cre under the control of the mu-opioid receptor promoter. We also saw that the majority of the Cre positive cells that were present and we were able to stain for also overlap nicely with our viral signature. Here's just some quantification of that. You can see we have very nice high expression selectivity for our virus across a number of these different areas in the mouse brain that are known to highly express mu-opioid receptor in them. So we extended this with some help from collaborators into other rodent models. There's a lot of work that's done in neuroscience research in mice, but mice are not always the best tool to use for a lot of different studies. There's a lot of downsides to them, and there's a lot of very complicated behavioral studies that you might want to do for addiction research or pain research that rat might be a bit more of a viable tool to use due to their higher capability for learning and memory-based tasks, as well as for other kinds of reward-based tasks. And there's also an interest to look into other animal models that might look at other modalities of addiction research. This Asian house shrew is actually unique because it's one of the few rodent, it's not rodent, it's a mammal, one of the few small mammal models that are actually able to have a vomit-based reflex to opioid-based overdoses in them, which is somewhat replicated by patients in the clinic too. If they have too much of an opioid, they can sometimes vomit to avoid themselves of that excessive opioid within them. So this animal is actually used for modeling NMS in that way. In the preclinical level. So with help from Dr. Matthew Hayes and Dr. Tita Borner, we just did a number of these same histological validation studies across a couple of the same regions that are also conserved in these other animal models. And we also saw the same kind of high selectivity for a virus in these animals too, indicating that it seems to have use outside of just the mouse field, but can be applied to a lot of these other very popular small mammal and rodent models that are used by a lot of researchers in neuroscience. So next couple of things we'll just go through are some of the functional validations that we did. It's nice to know that the virus is there, it's selective, but we also want to know that it kind of seems to target cells that are working in the way that we would expect them to work when you're engaging in either tests meant to look at addiction-based tendencies or pain-based tendencies. So what we did here was we took a version of the virus that expresses these dread receptors that I mentioned earlier. This is a modified GPCR that is very similar in its functionality to the mu-opioid receptor, again, which is a GPCR that drives more inhibitory-based signaling when it's activated. There are dreads that can do this same thing. There's these HM4GI dreads or HM3DQ dreads, which are inhibitory and excitatory, respectively. We put this inhibitory dread into the mouse spinal cord with the idea of targeting it to these mu-opioid receptor positive-based cell types to see if we could activate these cells more selectively by putting this agonist that should only go to these dread receptors when it's expressed, and see if that could replicate changes in different kinds of pain-based behavior in these animals. So here's some histology showing that we were able to get very nice expression and selective targeting of the virus to areas within the spinal cord where we should see the majority of the mu-opioid receptor cells, which is kind of up in these lamina 1 through 2-ish areas. You can see that there's a very nice high concentration of positive cells in and around there. When engaging in a couple of different behavioral paradigms with these mice, a couple of these tests, which are both mechanical, heat-based, thermal-based, and also chemical-based in nature, just to kind of drive different kinds of pain-like phenotypes in these animals. The long and short of this is what we were able to see is that in the animals that got the dread in them, the dread receptor in them, when we activated it with its cognate ligand, this thing called clozapine N-oxide or CNO, we saw that these animals had a heightened resistance to kind of showing this painful like phenotype when compared to animals who just got control virus injected into them, into the same regions of the spinal cord. So essentially what we were able to see is that the animals that had the dread and got the drug, they seemed to have a higher threshold for when they started to show painful-like behaviors across all of these different modalities, which in Von Frey involves lifting the hind paw up and kind of licking and attending it. The hot plate, usually you see animals licking and attending to their paws and jumping and trying to get out because they don't want to be on the very hot surface for a long time. And in the formalin hind paw experiment, this is also shown by injecting a noxious chemical like formalin into the hind paw. Usually very acutely, the animals will express a lot of licking and jumping behavior because they don't really like that in there. And then over time, you can actually see the prolonged kind of expression of these kind of same phenotypes in the mice, where they're kind of like limping, guarding their paw very heavily and closely, and in general, just kind of trying to lick it still to try and kind of mechanically gate that kind of pain and that noxious stimuli. And in general, we saw a reduction in all these behaviors in these animals, which was very positive to us. It seemed to indicate that the virus was, again, still targeting those mu-opioid receptor cells, and when activated, could replicate a mu-opioid receptor-like response without actually directly engaging the endogenous mu-opioid receptors that are on that cell surface. This was replicated using a different kind of agonist for these dreads too. This is called DCZ. It's kind of a more selective version of CNO. CNO has the nasty tendency to sometimes be just converted via drug metabolism into clozapine, which obviously we all know is not a great compound that can drive a lot of different psychoactive effects. So sometimes hard to say that what you're purely seeing is only due to the CNO drug itself and not the off molecule or two of CNO that's been developed. So this other kind of agonist was designed to kind of circumvent that issue. And we see a lot of the same effects replicate in these animals when using this other compound. So viable for use with both of these. The last couple of things we'll just talk about is we also tried to do this in a way where you could actually look and see the changes in the activity of different brain structures and regions that express these cells that have the mu-opioid receptor on them using these other tools. This is a tool that allows you to do in vivo calcium-based imaging, not at the same level of looking at individual cells, but looking at a change in bulk calcium signature. These tools, specifically called these GCAMP tools, are a class of molecules called GECIs, which are these genetically encoded calcium indicators. So essentially, this is a modified form of GFP that kind of works off FRET-based technology, where when the molecule is inactivated, it's bound to this CAMK2 molecule. When calcium is present, the CAMK2 will change structure, and it will allow the GFP to kind of come back together and form a functional protein. It will then fluoresce. And then in the absence of any calcium, when the CAMK2 is in its inactive state, there's no connection between these two portions of the GFP. So you'll essentially have very little GFP signature that you can see. So this has been very useful for people across neurobiology because it allows you to somewhat inelegantly, it's a bulk-based signal. It's not very temporally refined, but you can actually just see and get some indication that there's activation of specific neurons in these areas via proxy of calcium influx, which is required for driving action potential firing in neurons. So what we're actually able to see pretty nicely is that when we inject these animals with this virus, we can actually show that by activating a couple of different regions of the brain, we're actually able to see very nice and selective calcium-based activity. That's what this arrow is showing, this little boop uptick in calcium that we're able to kind of measure in these cells via the fluorescent proxy. This is the video. You can kind of see when this happens. I think you'll be able to guess when the animal is getting the stimulus and when that big bump of calcium is happening right about here. You'll kind of see the animal jumps very actively and aggressively. It doesn't like that air puff. This is injected into the CEA as well, an area that's known to encode different kinds of fear-based activities and anxiety-based information in mice. So this is a good kind of test to see if the GCAMP is working, and it does seem to be working pretty nicely in them. And we can get nice repeated activation of this GCAMP signature over time too. So what we were essentially able to do is now do this in the case of, instead of doing a air puff, we were actually able to give these animals either no morphine or an administration of morphine to see if we can engage opioidergic circuitry in them, and then apply them, administer them instead of like an air puff. We did a hot water droplet that's at a very high temperature that's noxious to them. Usually when the animals get this hot water droplet on their paw, they'll very quickly lift their paw up and start attending to it because they don't like getting burned. But we actually noticed was that while we were also able to see these nice signatures of activity in these mice, we actually saw a reduction in the overall number of them and the intensity of them in the animals that got morphine administered when monitoring the activity of these mu-opioidergic cells. So this is a quick video. It's kind of hard to see, but you might be able to see the animal lift its hind paw up when we give it the hot water right there. You can see he kind of lifted it up to his mouth and started licking it. And he'll do that a little bit more over time here, where he's kind of still kind of attending and guarding it a bit. By comparison, the animal over here has been given the morphine stimulus. Kind of see the hot water was applied there, and it doesn't really seem to care as much about it because it's gotten a pretty nice bolus of morphine and was allowed to wait for about 30 minutes for it to really fully engage the opioidergic circuitry. So here's just a breakdown of how that kind of all played out. When doing this hot water stimulation, we saw a slight decrease in the overall peak and overall number of these calcium-based responses in the opioid administered animals versus the control group, which seems to be indicating that there's a decrease in overall activation and engagement of these cells when morphine is on board, which is what we expect. And we can also monitor this in real time with this kind of technology. We also extended this to applications for addiction-based research too, by giving these animals a chronic morphine drinking paradigm over time, and then kind of doing an naloxone-based challenge to see withdrawal phenotypes in these animals. And the long and short of this is what we actually ended up seeing was that in the animals that did get either vehicle and no morphine administered over any time in their drinking water versus the animals that did have the morphine, when inducing this naloxone-based withdrawal, we actually saw a much higher increase in the number of calcium-based signatures that we were able to see, again, measuring from the CEA when compared to the control animal that you can see in the gray kind of trace there versus the red trace, which is for the morphine drinking mice. We also saw an overall reduction in the events that were occurring in the control animals and the number of global withdrawal-like phenotypes and behaviors in these mice too. So showing that this tool can hopefully have applications for testing in opioid-based models and chronic addiction-based models too. We did some ePhys too. As Julie mentioned, I did a lot of patching back in the day. We were having trouble getting Greg's rig up and running, but we were able to eke out a little bit here. This was essentially just showing that we're recording electrical responses from these neurons by doing an in vivo patch clamp on them. When we identified these mu opioid receptor positive cells via this GFP signature after viral injection to a brain region, we were able to record and see if there's a change or a decrease in the activation of these cells when we have an agonist on board that should reduce the overall activity of those neurons. And we kind of do see that. You can see these spikes are action potential firing events. They are reduced when you put something like DAMGO, which engages opioidergic circuitry, inhibits the overall activity of the cells. We can see that meted out very nicely here. This is just quickly going over a couple of different other viruses that we designed using this promoter region. This is essentially allowing you to just kind of target different kinds of populations of cells using a lot of the transgenic mice that are available as a tool for a number of neuroscience researchers who work with mice nowadays. That's more or less what we were showing here. And then lastly, we also made some of these capsid variants. There are these newer kinds of viruses that have been developed over the years by folks at Caltech and Vivian Grandinaru's lab. These allow you to kind of do a systemic injection of these viruses, and they can be restricted and sequestered either to the CNS more predominantly or the PNS. This is with the version that should go more selectively to the brain as opposed to the spinal cord. We can see that there's very nice expression of our virus across the entire brain after a single bolus injection. This is done retroorbitally, but in theory, you could do this intravenously as well. And lastly, we just wanted to see if we could extend this to higher order animal models outside of things that are used more predominantly in the clinic in the preclinical level and a lot of basic neuroscience labs. The mouse is pretty omnipresent in a lot of research. However, monkeys, which are a much better model and a bit more closely related to humans, it's a lot harder to do some of these studies, especially at the selective cell population level, because there are no transgenic monkeys, and there probably never will be, because there's probably no way that any regulatory agency will allow you to make a transgenic monkey. So using these viruses is kind of our next best thing. So this was done in collaboration with Michael Platt's group and a very talented postdoc in his lab who's now going off to Quebec City to start his own research group, Sebastian Tremblay. He helped us do all these intracranial injections into the macaque with both of these viruses. You can kind of see here's on the right is just showing where all the viruses were targeted to in the monkey brain. This was a lot of the same areas that we initially did some of this pathfinding for in the mouse, just into the conserved structures there in the macaque. And so again, we saw very, very nice expression of our virus, which again, in no small part due to Sebastian's expertise and talent as a surgeon. And we also showed that this was actually highly selective in the macaque brain as well, with the majority of cells that were transduced by the virus showing some signature of OPRM1 mRNA in them as well. This was also something that we did in culture too. This is just a final point, just to kind of think about drug design and drug development screening applications of this tool. Obviously, a lot of times you'll have compounds that you want to screen to see if they're more selective for a specific subtype of cell. This is kind of our way of showing that maybe you could do this kind of more in vitro at a high level throughput screen by making a culture of cells that might be more pluripotent. There might be a very different variety, or sorry, it's a more heterogeneous population, but if you want to see if you could selectively show changes in fluorescent signatures, something like that, when you do a screen in these mu-opioid cells more selectively than other cells that might be in that homogeneous mix of cells in your dish. Working with Will Renthal over at Brigham and Women's and Harvard Med, we were able to show that we could actually get very nice expression of our viruses in human-derived pluripotent stem cells that were kind of differentiated into nociceptors, as well as in mouse ganglial cells that were cultured too. So again, showing very high selectivity and good expression of the viruses in dishes there. So that's pretty much it. This is just a summary of kind of what we did and found in our paper and what we published on, showing that we made these tools, we validated them, and we were hopefully able to convince people that they're really useful applications to folks who are in the preclinical field, working across a number of different model systems and trying to study both pain and opioid addiction, and trying to find different ways to kind of more selectively show that you can modulate these cells to kind of study them from a different variety of angles, be it anatomical, functional, other things like that. And just the final thing on future directions, our lab is also now still working with Dr. Tremblay and Dr. Platt and trying to do some more of this work at the level of the macaque. In this case, putting one of our dread expressing viruses that's more selectively targeted to the mu-opioid receptor into these cells in the monkey brain, and then hopefully trying to get approval to do some behavioral testing in the macaques to show that the effects I mentioned in the mouse that we noted when using this injection in the spinal cord might be able to carry over into the macaques in a higher order model, where we show that by modulating those cells, by giving that selective agonist to these drug expressing neurons, we're able to reduce maybe the pain or the addiction-like phenotypes that you could see in these animals without, again, binding up the actual cognate receptor. So in theory, the ability for the animal to sense pain, do other things like that should be conserved, but we're just modulating the activity of those cells a little bit over time here and there. So that's it. Again, just a lot of thanks to everyone who made this possible, both in Dr. Quarter's group, Dr. Platt's group, all of our other collaborators across a number of different institutions outside Penn as well. Of course, the T32 to Julie and Anna Rose for making this all possible, allowing us to have some of the funding to pursue and get this all the way to publication. And if there's any questions, I'll take it, but I know that we're kind of pressed for time a little bit too. So just let me know. Happy to chat anytime offline or just via text or email.
Question while somebody's thinking about their question. I think just this is so elegant in terms of showing the impact on the, I'll call it the aversive side of the motivational coin. Do these same viruses, these interventions that you're doing, have impacts on competitive models?
So we didn't specifically look into that yet. It's possible.
If there's any symmetry there, or if it's just that it's really exclusively on the.
So, I mean, there have been a number of groups that have been trying to develop tools like this to go more exclusively after things like dopaminergic cell types, which might have more carryover to appetitive-based models or other kinds of addiction-based models as well. We mostly looked at this just in modalities of pain and opioid-based administration, chronic opioid consumption as well. But in a way, that's a appetitive-like model too.
I'm just curious. Okay.
Yeah.
I'll shut up. Anybody else? Go ahead.
So, but I guess technically these will go into a variety, like all the different GABA cells, for example, that have containing receptors, even though we know there are subtypes of GABA.
Yes.
Is there any attempt to sort of start to localize them within neurons?
So in terms of a specific subpopulation of opioidergic cell types, that's kind of why I breezed through it, but in looking whether or not this can be done in complement with other transgenic animal models that are present. So what we showed in that one slide was that using an intersect-based approach, you can actually get expression of these viruses in either other Cree-based lines that are maybe Cree for a certain GABA or certain, something that's more glutamatergic. So in theory, if there's a Cree line that should allow you to more selectively target a subpopulation, let's say of opioidergic cells that has that signature in it, we've shown that you can, in theory, use these Cree-based viruses or Phlox-based versions of the viruses we've made to drive a selective expression in there as well.
Questions online?
Ask your online folks if they have any.
If anyone has any, let me know. If not, I will be happy to move on and is Nathan next or is it Cindy next? Cindy next, okay. I think that's it then. So I will exit out. Guys, thank you very much as well for your time. And yeah, please let us know if you have any other questions about this. I can direct you to Greg or myself to kind of field some of them.
So I'm going to be shifting more to the clinical side and sharing I'm going to talk about the interrelationship between nutrition and substance use. More specifically, I'm going to be sharing some of our recent data looking at the effects of ketosis on responses to alcohol. To give a brief overview of nutritional ketosis, it is a metabolic state that's characterized by increased level of ketone bodies and is usually achieved through the breakdown of fatty acid when glucose is not readily available, such as during starvation or adherence to a ketogenic diet. However, there are also several commercially available products that is able to exogenously increase levels of ketone bodies without adhering to a ketogenic diet, which is very restricted. It's usually characterized by 5 to 10% of carbohydrates. So if you eat a banana, you're out of ketosis. So this kind of offers an easier way to increase your level of ketone bodies. Both preclinical and clinical data have evidence the role of ketosis in dampening alcohol withdrawals. So this left panel showed a mice study in which they give mice, along with alcohol, a ketogenic diet, or a ketone supplement. And they found that the ketogenic diet as well as the ketone supplement decreased alcohol withdrawal, which they measured using handily induced convulsions. So they did area under the curve up to 3 hours after alcohol abstinence and then up to a whole day. So it seems like the ketogenic diet has maybe a stronger effect than the ketone supplement. And previous work by my mentor Corinne at NIH demonstrated that the ketogenic diet is effective in reducing the need for benzodiazepine for the management of withdrawal symptoms in inpatient with AUD during detoxification. However, the effects of ketogenic diet versus ketone supplement on alcohol bioavailability kind of is very conflicting. In mice, this study showed that there was no effects of the ketogenic diet on blood alcohol levels. However, they showed that the ketone supplement was effective in reducing blood alcohol levels. However, work done by Corinne at NIH, this is in rats, showed that a ketogenic diet resulted in 5 times higher blood alcohol levels in rats that were alcohol vapor exposed. So what is the effects of the ketone supplement on alcohol responses in humans? So this is our study looking at the acute effects of keto supplements on responses to an alcohol challenge in healthy volunteers. This was registered on clinicaltrials.gov and it was recently accepted for publication last month. The primary objective is to examine the effects of ketone supplement versus placebo on breath and blood alcohol response. And the secondary objective is to look at subjective reports of intoxication and alcohol craving during an oral alcohol challenge. Just one thing of note is there are several different commercially available ketone supplement. The previous my study that I mentioned, they use a ketone ester, which breaks down into 1,3-butanediol and beta-hydroxybutyrate. All of these ketone supplements break down into beta-hydroxybutyrate, which is the ketone, one of the ketones that's found in our bloodstreams. For our study, we use this product called Kinetic. It contains 25 grams of total ketones that is made of 1,3-butanediol and beta-hydroxybutyric acid. This compound that we gave has a total of 150 calories, and our placebo, unfortunately, it has sugar, which have been shown to also affect alcohol metabolism, and that it would dampen blood alcohol response. And also this has 57.2 calories.
Can I just ask, what do people normally take this supplement for?
So right, So they're marketed as like for workouts. So people take it kind of to help boost their like workouts. There's some studies showing that it might affect cognition, but I think at least on their website, they are kind of marketing as like workout supplements. This is a crossover study, a two-way crossover study with greater than 1 day washout period. So the participant comes to our study center after overnight fast. We give them a standardized meal. An hour later, we give them either the keto supplement or the placebo, followed by alcohol approximately 30 minutes later. We collected blood levels of glucose and BHD, blood alcohol levels, as well as breath alcohol levels and certain questionnaires. These are the characteristics of our participants. They're about 30 years of age, 3 females, 7 males, normal BMI, 1 African, 1 Asian, 8 whites. And then these are the education, and we require that all participants drink at least 2 drinks on a single day within the past month, so that they have some kind of experience drinking, so they don't be really surprised when we give them vodka. So this was our study finding. We found that as expected, the ketone supplement lowered blood glucose levels as well as increased blood levels of the ketone body BHB. We saw that this administering ketone supplement significantly dampened both breath alcohol as well as blood alcohol levels. And so the breath alcohol levels in the people that receive the ketone supplement is about half of during the placebo arms.
And Cindy, that was novel, right? That finding?
Yeah. So yeah, this was this was the first one that was done in humans. Just one thing to note that we send the blood to Hub for analyzing blood alcohol levels. Hub has a cutoff of 10. So for those that were below detection, we just arbitrarily set 5. We use a drug effect questionnaires, which has 5 questions. Looking at feeling the effects of alcohol or feeling high. These were the first 2 questions. We did not see any difference between the ketone supplement and the placebo. With regards of disliking, liking, and wanting more, there was a significant effects of the keto supplement where they felt higher dislike, lower like, as well as lower wanting more of what you have consumed. For liking more, there was a significant difference at baseline. So this was before alcohol administration but after the ketone supplement. And then this effects of the ketone supplement kind of disappears when controlling for this baseline measure.
Can I just confirm, this is not mixed with the alcohol, correct?
Yeah, so this was given half an hour before the alcohol. We also measured alcohol craving. There was no significant difference. Alcohol is associated with a biphasic response of stimulation and sedation, but there was no effects of the ketone supplement on the sedative or the stimulative effects of alcohol. Before you go and buy a keto supplement, before you drink alcohol, I just want to share some of the adverse effects that were reported in our participants. There were 3 participants who reported some kind of gastrointestinal discomfort, and 1 person dropped out. And then there were several cases of hypoglycemia, at least at some point during the study visit. So this product that we use is a mixture of 1,3-butanediol and beta-hydroxybutyric acid. It also has a copious amount of this artificial sweetener allulose. So we invited back the participant to do 2 more arms of the study. looking at the effects of the 1,3-butanediol and allulose. We couldn't do the beta-hydroxybutyric acid because it is not commercially available. However, we have some collaborators over at NIH who is currently studying this in mice, rats. So it's basically the same study design. We didn't we collected less blood alcohol level just because we didn't want to sick the participants too much. So we showed that there was a treatment effect on glucose. This keto supplement increased levels of BHB. This 1,3-butanediol did too, but less so than the kinetic. And surprisingly, we found that only the ketone supplement was associated with lower breath alcohol levels. And then, so the effects that we saw was not due to the artificial sweetener, as well as 1,3-butanediol. So there may be evidence that different ketone supplements have different effects on alcohol. But our collaborators looking at the effects of the other ketone body that's in the supplement right now in rats. Also, we're just beginning to try to understand the mechanism through which the ketone supplement decrease blood alcohol levels. So our collaborators at NIH, they studied this by giving the rats ketone supplement or placebo, and then they gave the alcohol either intraperitoneally or through oral gavage, and then they collected blood alcohol at various time points. So they also found that this ketone supplement dampened blood alcohol levels, however, only when alcohol was given orally, but they did not see an effect when alcohol was given IP. We're kind of we kind of think that this is because that the ketone supplements dampen alcohol absorption and does not really alter alcohol metabolism. That's our working theory, and we're trying to figure out more in detail of how this ketone supplement works. So kind of as a summary of our study finding, we found that the ketone supplement, when given before an alcohol challenge, dampened blood and breath alcohol levels, decreased liking and increased disliking the effects of alcohol, and decreased liking more of what was consumed. It does not alter feelings of sedation or stimulation, or did not affect alcohol cravings. These effects were not attributable to the sweetener allulose or 1,3-butanediol, and preclinical data suggests that it likely decreases absorption, not metabolism. There are some limitations and future directions. The human studies, we use a relatively low dose of alcohol. Maybe that's why we weren't able to see that big of a difference on subjective ratings of intoxication. Our placebo was not the best placebo because it contains sugar and is also not calorie matched to the supplement. We saw that there's a high inter-individual differences. So with some participant after the ketone supplement, their breath alcohol levels is like 0 the whole time, whereas other participant, the ketone supplement didn't affect them as much. So it'd be interesting to know why, and what is the implication of the ketone supplement's effect on alcohol translate to potential treatment for AUD and alcohol craving. Do variant types of ketone supplements have variant effects? And then going back to kind of more introduction, we saw that the ketone supplement decreased blood alcohol levels, whereas the ketogenic diet increased blood alcohol levels. So looking at the differences between the ketone supplement and the ketogenic diet. And does ketogenic diet and keto supplement produce neurochemical alterations that have implications on subjective responses to alcohol? So we are currently recruiting for a new study that can more aim to address these last two questions. This is a study rationale and aim. As I previously mentioned, the ketogenic diet and the keto supplement have differential effects of alcohol. So we want to see what is the effects of ketosis that's induced by ketogenic diet versus keto supplement on alcohol intoxication and metabolism. Ketones have also been shown to alter the carbon flux in the TCA cycle and alter levels of glutamate and GABA. So what is the effects of ketosis on brain metabolites? And GABA and glutamate may have implications on subjective responses to alcohol. So to explore the relationship between GABA and glutamate on subjective responses to alcohol. This is our experimental design. It's going to be a 3-way crossover study in which the participant received 3 days of ketogenic diet, 3 days of standard diet, or standard diet plus 3 days of daily ketone supplement. We know that 3 days is probably a really short time period, but we also want to see it. But it's hopefully it's short enough for the participants to adhere to the diet. So they'll receive the diet for 3 days, and then we'll do a alcohol lab study visit on the 4th day. So for the lab day, they're going to receive a breakfast, and we're going to measure GABA and glutamate at the 7 Tesla, and then they're going to be given alcohol, and we're going to measure breath alcohol levels, different questionnaires, and we're also going to be looking at their motor and cognitive tests. So we just started recruiting if anyone wants to volunteer. And then that's, so thank you for listening. And I'd like to thank a lot of our collaborators, Corinne, my mentor, as well as our clinical research coordinators. And I'll take any questions.
Oh, go ahead. So what, first of all, did you look at any sort of liver function or liver enzymes or anything like that? I guess my question is in the big picture, is the goal to, just for people who have an alcohol use disorder, or, I mean, is it for people who want to drink, but then if they drive, it will reduce their blood? I mean, you know, that's terrible, obviously, but it would work, right?
That's what the company is really happy about, not saying that. That's a market.
No, yeah.
But I think for that, I think we also want to give the alcohol first and then the ketone supplement, kind of alluding to, oh, I drink too much, I need to drive home, let me clear this out. But we haven't really tested that yet. And I think with regards to alcohol use, we did see that there is a increase for disliking the effects of alcohol. It's also it's not the most pleasant thing to drink. It kind of, so some of our participants, I don't think they like the feelings, like maybe they just don't want to drink, but I don't know if that's a transient effect or can have implication for treatment.
We're starting a study now in an inpatient setting to get this kinetic during breakfast, lunch, and dinner, in addition to their regular, to see if similarly to ketogenic diet, whether it decreases alcohol withdrawals. But that is, of course, the absent. There's no alcohol.
Right.
We're already at 0, and we give it to them.
Yeah, that's what I was just like, is it kind of like the anti-abuse approach, or is it, you know?
Well, I mean, it's intriguing. You get a big lockdown on blood levels of alcohol. I think that's just obviously companies interested. Everybody's just curious about that. That's a big finding, in addition to wanting the withdrawal or the other things. So, and I'll be interested to see with your new stuff, the behavioral test that you choose for looking at alcohol impairment. What would you choose?
We use it's called a PEG groove board. So it has like it's like a key, except it has different orientations and people put the key in the hole and see how long it takes them. And then we're using go, no go to look
Trying
at,
to
you
get
know.
back into your apartment in the evening. Partying, and you're kind of like, I know this key is supposed to work.
Yeah.
Not that that's in. Seen it on television. It's very interesting, and it also speaks to the, you know, serendipitous findings. I mean, when we walked in, it was mostly focused on the withdrawal, right, and the negative components of withdrawal.
Yeah.
And here you are with new findings.
Yeah.
Interesting. Thank you very much.
Hard to believe we're at the very last presentation for today. We're very happy to have with us Nathan Hager. Nathan actually returns to us after having been a valued and special research assistant some years ago. He went away, got his PhD in clinical psychology at Old Dominion, and actually comes now to us highly skilled and already contributing to our projects in the last several months, but he's the newest addition to the T32. So he's going to tell you some of the things he's been doing. He's plunged in, he's up to his elbows or higher up and work of different sorts and already forming other collaborations. Nathan, welcome.
Thank you. So I'll be talking to you about,
Excuse me, did we- Okay.
oh, I need to share my screen. Yeah, good point. Thank you. So I'm going to be talking about a study, some data analysis I've been working on and will continue to work on, and some things that are coming up in some of the projects I'm working on. But all of this is related to looking at how mechanisms in the brain and cognitive function influence and contribute to opioid use or maintain opioid use in people with opioid use disorder. So I'm going to introduce the concept of brain network segregation. This is a measure to try to compare how different parts of the brain are connected or with each other, either within a network where the brain is working together for a single function, or with a connection to other networks where they're usually more sparsely connected, but still connected to networks outside of the nodes outside of the network that they're in. So network segregation is comparing whether if you have more segregation, then you have a increased within cluster communication and decreased between cluster communication between different networks. So for example, nodes within the default mode network are going to be more connected than to nodes within other networks like the visual network. And so you can see one way that these networks have been described as in broader terms is as association networks or sensory motor networks, where the sensory motor networks are like visual, auditory networks that work towards motor function and senses, and then the association network are cognitive, integrative kind of networks so we'll be looking at those separately. And this is how we calculate network segregation. It's based off of graph theory, but it's a more, the actual calculation of it is easier than other methods of graph theory that sort of do the same thing as network segregation, or they're theoretically similar, like modularity or participation coefficient. They work and they gather the same sort of information and they actually correlate together. But in network segregation, we're looking at the correlation of nodes within the network and subtracting the connection of the node to other nodes outside of that network and dividing that by the within network connection. So basically, how much are these nodes connected within the network versus outside the network? And so brain network segregation, why are we interested in looking at this? So it correlates, I already said that, but network segregation actually does a better job at predicting age-related changes in and how the brain is connected to it, to different areas or within itself and outside networks than the modularity and participation coefficient that I mentioned. And so it possibly could be better at predicting clinical outcomes as well. And so network segregation is theorized to help the brain work more efficiently and through having functional specificity within these networks. And a paper by Manza and that one of my mentors, Corinne, participated in did show that people with lower segregation or lower segregation was associated with greater age-related decline in glucose metabolism. So indicating that segregation is important for efficient brain activity. So like from a clinical perspective, like I mentioned, lower segregation is associated with higher age. So as you age, your brain becomes less segregated, more connections between different networks, so possibly becoming less efficient. And then also it's associated with worse cognitive performance. Recently, our lab found that people with alcohol use disorder actually have a lower level of segregation, network segregation. And so that paper is under review. And so my research question was, how does this look in an opioid use disorder? And I was looking at it rather than opioid use disorder versus not having opioid use disorder, looking at a continuum along severity of opioid use disorder. So both looking at the association networks and the sensory motor networks, and then also seeing more specifically, are there networks that are driving the effect of opioid use disorder severity on segregation. And looking at more specific networks is not something that's been typically done in the literature, so could be something interesting. So I did this looking in a sample that was collected here. I actually helped collect the data when I was a research assistant many years ago. The CURE study using data that Dr. Lengelben headed up in collecting. And so these are individuals who were treatment seeking to receive naltrexone for their opioid use disorder. And this was data collected prior to receiving treatment. And using resting state scan data. So I used a measure of opioid use, a measure of drug use severity from the ASI, and did hierarchical regression analyses to predict network segregation, also controlling for age and for motion, which was quantified here with the mean frame-wise displacement measure. And this is important because it has a big movement during the scan, really impacts functional connectivity. And so we're controlling for that here. So within the association network, there's networks that are related to cognitive function, integrating different functions. We can see mean frame-wise displacement was significant, so we're good to include that as a covariate here. In my second model, I added age and the ASI drug severity score. So for this, we can see that this is a marginal association with age. And as far as age, predicting decreased network segregation in the association network, but we have a significant association with drug use severity. So as drug use severity increases, the network segregation decreases across these 40 participants in the study. And the partial correlation here is negative 0.48. I also included the interaction in the next model between age and ASI drug severity, and that did not improve the model. So we're sticking with the model number 2. So here you can see graphed the association network segregation on the y-axis with ASI drug score. So that is a correlation of negative 0.29. Like I said, the frame-wise displacement contributed to network segregation, as did age somewhat. And so I partialed out those two covariates, and you can see the effect more strongly, and this is reflected in that correlation being significant. So as we look across age, or look across the drug use severity, we see network segregation is decreasing. So I want to look at also the sensory motor networks, and findings were a little bit different. Age has a stronger effect on the sensory motor networks. So as age increases, the sensory motor network segregation decreases. And then we also see a significant interaction though. So the model improved when adding the interaction between age and ASI drug severity, even though drug severity did not have its unique effect on sensory motor network segregation. So what this means is that the effective age depends on drug use severity, or the effective drug use severity depends on age. So we'll break that down a little bit. So I broke it down by looking at age and the different, looking at different points along age. So these are the effects at the -1 standard deviation, which is the green line, the mean of age, which is red, and then the +1 standard deviation of age, which is that purple color. So you can see when the participants are younger, as drug use severity increases, the sensory motor network segregation decreases. But for people who are older, it tends to be the opposite direction. So increasing drug use severity actually increases the segregation. So the effect of drug use severity is different at the different age levels. To break it down a little bit more, I looked at, so where is this effect actually significant? So I did a Johnson-Neyman plot to model where the effect of ASI drug severity was significant, and it was really, you could see these opposite effects at the two ends of age, but the upper end of age was just 4 participants, so I didn't put much stock in that. But the lower end, we have, starting with people who are 23 years old or younger, we see this effect, and that's what you can see plotted on the right here. The correlation is -0.55. So for the younger people, people who are less than 24 years old, as drug use severity increases, their network segregation within the sensory motor networks decreases. So perhaps, and this is recent, so this is drug use in the past 30 days. So that is capturing here. So recent drug use severity is contributing to the sensory motor network areas communicating more with other areas, and there's bleed over in the functional specificity of these sensory motor network areas. So I tried to graph and show this a little bit, and also understand it a little bit more. So you can see on the left side, the low drug use severity group and the high drug use severity group. I split them by plus or minus 0.5 standard deviations. So what I got out of this was that you can see the biggest difference I could see here visually, and this is a sparse network, so it's not including all the information that's included in the analyses. So take that into account. We're sort of approximating what the analyses found here. But I thought this frontoparietal yellow group here looked more connected within itself and less connected with other networks than in the high ASI. So you can see it's a little bit more centralized than in the highest AI group. And the lowest AI group, I also thought that the black salience network looked more centralized than and more spread in the low ASI and more spread out in high ASI. So I thought maybe there could be something to look into those different networks. So I did the analyses again, expecting to see that's where we would see those two networks driving the effect. And so this was more exploratory, just trying to see what was driving the effect here. And that's what I found. Those are the two networks that were driving the effect of the network segregation changes related to the drug use severity.
Did the connectivity within the networks, it looks like all your lines are the same sort of strength. So sometimes you see like, you know, the lengths or the edges.
The edges, yeah, so the edges are actually reflecting the strength of the correlations. Yeah. Yeah, it can be hard to tell, yeah. And so in the frontoparietal network, which is associated with task switching and integrating emotional cognitive information, we see the effect of ASI drug severity on network segregation. So as the drug severity increases, there's a lower network segregation, lower specificity within this cognitive network. And then we also see in the salience network, which is associated with detecting important information, integrating emotional information with sensory information. We see the same thing as drug severity increases, network segregation decreases. So those are my network segregation findings. Initially, I'm looking into also explaining this to other drugs that we have data on, cannabis and cocaine use disorder, to see if there are any differences across these samples of people. And also using network segregation to look at treatment and how it might be related to treatment, predicting treatment. And so that's what I wanted to talk about in a second, but yeah, we can pause for a second.
Do you have genetics with these opioid use guys?
Do we have genetics?
Yes.
Yes, we do.
Yes, we collected blood samples and the cure, and we've been a lot of work that we did with SNPs when SNPs were still more in fashion than they are now.
They still are in function.
Whether we have some samples and we have whatever was
I
analyzed.
believe, you know, we kept the extracted DNA so that then you could, for the price of a kit with reagents for a certain SNP, you could run it. And as far as I know, that's still the deal. Should double check them.
Because which OPR1, the
Yeah.
A1ATG,
Yeah.
would be very interesting to know. I'm sure it was. I would have been surprised if it wasn't here.
Okay.
That would be interesting to see how that layers on top of the network.
Yeah. And so I'm also, any other thoughts or questions about the network segregation?
Say, walking in the door, Nathan, would your thought have been that greater severity, lower segregation, meaning more spread?
So that's what I was expecting, just because of the AUD results, and also because OUD is related, can carry some cognitive impacts where there's poor cognitive function, and that was something that had been found as related to lower network segregation.
Sure.
So it could be that network segregation could be a player, like impacting the cognitive function, and actually could be one of those brain mechanisms that I would be interested in looking
Yeah.
at.
I mean, we've talked about, and with no small amount of energy, Nathan has gained access to the ABCP data set. So one of the things one could do is, as you thought, maybe look in those young kids for vulnerability signatures
Mhm.
among these networks.
Right, and
Do you
then
have
as
thoughts about
substance.
this you'd like to share?
Yeah, and then as substance use emerges in that data set, it's a longitudinal data set, we could see changes perhaps in network segregation.
Or anything that predates it, right? And maybe
Yeah.
interacts with it. Like,
Exactly.
one of the interesting things is, since we usually, without the ABCD, we wouldn't be able to know when we see our adults what came first. You know? And we keep trying to use, try to be careful about using association language,
Right,
because we don't usually have a causal model. But the ABCD will allow you to test some of those things.
Right. And speaking of causal models, this is I wanted to sort of talk about the elephant in the room. The elephant in the room about functional connectivity is that we're looking at Pearson correlations between these different nodes, right? So that's functional connectivity, but not effective connectivity, where effective connectivity is what's the direction of this relationship? What is one node predicting activation of another node? Are they just going at the same time? Are they simply correlated? So another direction that I'm working on with a collaborator who has been developing causal discovery analysis for use with MRI data, resting state, and also functional MRI data. His name is Eric Rawls at the University of Minnesota. We are hoping to be able to look at and create a more sparse network of these connections by weeding out the connections that are spurious in reality, because they're not actually causing each other. And so the model uses machine learning to create a connectome for each subject, and then finding what is the most plausible directional connectome for each node. And so instead of so if you had if you had a network where on the left here, A causes B causes C causes D, we would want to know that. But by doing the Pearson correlations of functional connectivity, we get this on the left. We would think that D is connected to A. We would think that D is connected directly to B, but that might not actually be. Those might be spurious connections. So we're going to try to use causal discovery analysis to create a more sparse and also more helpful network where we could know, like, what are areas of the brain that we would want to target to impact other areas, maybe with stimulation, maybe with medication.
But can you just do control theory for that?
Tell me more.
Well, I'm just wondering, like, basically, you're just extrapolating from a single point of an fMRI, right? So I don't know how you would get directionality from that.
Right, yeah. So that's the so it's this causal discovery analysis uses a method that Eric has developed that I'm not as familiar with, called Gango. And it's is machine learning where they are searching for what are the plausible connections. It's doing it over and over again. And then it's estimating the effects of those using a structural equation model. So I'm not sure how to compare it to what you were mentioning.
So that was just when you had mentioned about like what brain regions might be more important. It's you sort of network control theory where you would take out one brain region.
Oh,
and see how
okay.
the others react.
I
and then
see.
You can assess which is
Right.
which one is the one that drives, from
Yeah.
state A to state B.
Yeah, so this would not be experimental at all. It would be modeling, yeah, computational, yeah.
It's interesting.
Um,
It's hard to get people to sign up for that.
yeah. Um, So another application is within the treatment studies. So we have a recently funded grant for psilocybin intervention for OUD, and one of the exploratory aims is to look at network segregation as a predictor and as a possible indicator of change across the treatment. So there's evidence in depression study that modularity, which I mentioned before, is related to segregation, actually reduced across the treatment. So that was surprising to me, given what we know about segregation in age and segregation in cognition. But the psilocybin, the mechanism might be that psilocybin is actually increasing connections within the brain to create a more neuroplastic, more flexible brain that can learn across the course of treatment to help people change their behaviors, to think differently.
Not just an old brain.
Right, yeah, it's not just creating an old brain, but it's actually maybe hyper connected, maybe more. And so, but that would be a question, that's a question, is psilocybin, could it normalize segregation within the OUD brain to help treatment, or could it actually create more connection across the brain that we're calling pathological here, but actually in the context of treatment, where they're learning and trying to change, could actually be beneficial.
It's actually super interesting because the brain network segregation signal correlates very much with brain glucose metabolism. Both really go down with age. And psilocybin has been shown to directly influence glucose metabolism. So that might be
Oh, yeah, interesting.
it's okay.
Yeah.
Borrow a pun.
Serotonin receptor, yeah.
So I'll just quickly go to another treatment related application. This is I was going to maybe give a little bit more background about flexibility, which is something that we will be looking at in the psilocybin study. So flexibility across different domains. So the brain, how does the brain support flexible behavior where you are responding appropriately to whatever changes are going on in your environment, and adaptively changing your thoughts or changing how you feel, changing what you do in order to survive in your current environment. So that could be parts of your brain that support adaptive behavior or changing the way that you think. It could be physiological arousal flexibility, so heart rate variability, the measure of your heart changing across time, a heartbeat changing across time, where it's not just a steady beat, but it actually responds to the environment. It might be related to flexibility in this way. It actually has been shown to be related to flexibility and your thoughts and your emotions. Cognitive flexibility, switching between different cognitive tasks and different rules when they change. And then psychological flexibility, sort of a way of regulating your emotions by changing the upregulating, downregulating emotions. And so I'm looking within all these different levels of flexibility, and we're looking at that within the psilocybin study, and also within a study that I've proposed and submitted an ITMAC grant last Friday. I think I'll just skip this because it gets a little bit in the weeds, but I'll talk about, so we talked about psilocybin, and I'll just talk about this other treatment. So memory flexibility training. This is a cognitive training that people do on their own in a workbook, where you practice recalling more positive and specific autobiographical memories. Autobiographical memories are important because they support our our concepts of ourselves. They support our ability to plan goals and motivate future behavior. They support our ability to regulate our emotions as we recall things that were positive, or we focus in on specific kind of information from our past to help us understand and deal with our present. And so people with OUD and across psychiatric disorders have difficulty with this, especially with retrieving specific positive emotions or positive memories or memories that can help them deal with the present. And so this MEMFLEX, memory flexibility training, has been used in depression and trauma to help people deal with this deficit. And so I'm hoping that that I'll be able to get people with opioid use disorder to buy into this and practice actually retrieving memories that are positive, but not related to drug use, but related to other things to help motivate reward behavior, but not drug use behavior. And so the aims of the study are to assess autobiographical memory and also reward flexibility, which is a term that I made up, in which you can regulate your desires and your motivation for natural and adaptive rewards, while sort of down-regulating your motivation for drug-related rewards and your response to drug cues. And so in that way, you're being flexible in how your reward system is responding to your environment. And then also we'll be doing FNIRS to assess the prefrontal cortex, assisting people in these skills, and testing how MEMFLEX might improve these dysfunctions. And then predicting changes related to autobiographical memory and reward flexibility from heart rate variability when doing these cognitive tasks, with the idea that higher heart rate variability assists with your ability to respond to changes in the within these cognitive tasks. And so trying to understand the mechanisms of the cognitive deficit and how that interacts with reward deficits as well. So that is what I've been working on and hope to continue working on. And I really appreciate all the data that I've been able to collect in the past and use now. From Daniel and Anna Rose and Corinne with helping me sort of find and latch on to some things that are really interesting here in my first 7 months.
We're very grateful to have you here, Nathan, because basically having you here makes such good use of these data, right? So especially for postdocs working in the clinical domain, prospective data collection is just glacially slow. So it's really great to have some existing data sets where you can have hypotheses and build models and test things without having to collect every little bit of data yourself. On the other hand, you do want to have some projects where you collect every little bit of data yourself, so you get used to how you measure things, what works.
Exactly.
And Nathan has plunged in, as you can see, wholeheartedly.
Right.
And
Yeah, so my goal is to do clinical trials and to incorporate the more cognitive behavioral treatments that we don't do so much in our department or center, and target the same kind of mechanisms, but do so with less invasive and maybe more friendly treatments to, especially with like MEMFLEX, where you're doing something on your own at home, and see how these things might be targeted, and also work with the medication treatments in conjunction.
Yeah, that's not an either or.
Yeah.
But thank you so much, guys, and I'm sure we've been thinking about hard competition. Thank you.
Bye,
everybody.
thank you.
Online.